For today's 3-minute paper, we'll be taking a look at a case study on SARS-CoV-2 evolution for a patient that is immunocompromised and chronically infected with SARS-CoV-2. In this study, the researchers were interested in the effect of treatment on the rise of new variants in response to certain treatments. In the study, researchers used a variety of patient sequences, including the patient of interest, X1, in a phylogenetic analysis against the Wuhan HU1 reference sequence, and saw that there was a considerable diversity for the patient of interest compared to other patients and existing sequences. With this in mind, the researchers wondered whether certain treatments throughout their course with treatment of remdesivir, RDV, and convalescent plasma, CP, was associated with the rise of different variants and changes in viral load throughout the infection period. As you can see, under prevalent variant treatment response, after treatments with remdesivir, there was not many changes in new variants rising from the wild type that was associated with the patient. But once convalescent plasma was given, there was different changes in the viral load of different single and double variants throughout the remaining course of treatment, with significant information associated with the rise of the D796H and the Delta 69, Delta 70 deletion mutations arising after convalescent plasma treatment. With this in mind, the researchers suggest that there may be linkage associated between certain positions and rise of those mutations against different treatments. To better understand the response to convalescent plasma, researchers created a pseudovirus with luciferase to track changes in sensitivity to the convalescent serum. And in that experiment, they found that with changes in infectivity measured through the change in luciferase compared to the wild type, different variants had different infections compared to the wild type, as shown with D796H showing a lower infectivity. But importantly, the D796H sensitivity to the convalescent serum was much lower compared to the wild type. And together, in the double mutant, once examining the convalescent serum of the patient after treatment at day 66, they could see that there was still a very low sensitivity to the convalescent serum. Overall, the researchers know that only one patient does not confer this as evidence for a wide range of SARS-CoV-2 evolution being induced by various treatments, but with the advent of immunocompromisation in the patient, they suggest that further studies in clinical trials and surrogates would provide greater information to understand how these variant evolution evolve with various treatments that are available currently.